Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you. Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. Don't forget we have a new book out called Understanding Viruses. You can find it by going to Amazon and typing in Finding Genius. It's the result of interviews with 25 top virologists, asking them very difficult questions about virology. So check it out there. Today, my guest is Nikolaos Robakis, the AP Slaner Professor for Alzheimer's disease research at the Icahn School of Medicine, Sinai. So, Nikolaos, thank you for coming. How are you doing?
I'm doing fine, thank you.
Well, what got you interested in it at such a young age?
Yeah, I was very young. I was working initially with bacteria and viruses. And then there was a move to start also applying molecular biology and genetics to try to understand human disorders.
Okay.
And that's how I got interested in Alzheimer's, because I was also working on, mad cow's disease, mad cow's disorders.
Yeah, I've heard about that. It's called prions.
I was also working on that. And slowly I dropped that and I kept working on Alzheimer's disease.
Well, it's interesting you should mention that. Has anyone ever found prions to be associated with Alzheimer's disease or no?
It's still unclear. There are some similarities, like the deposition of amyloid, that in some cases we see that also in mad cow disorders, that we call it prion disorder. Okay. But there is one fundamental difference, very important difference between the two diseases. One is transmissible and the other is not. And the transmissible is the prion disorders can be transmitted. But thus far, we have no evidence that AD is transmissible.
Okay.
So that's a very important difference between the two disorders.
Right. Does anyone know how Alzheimer's disease starts? I mean, you've been doing it for, like you said, 40 years. What have you discovered about it that is very significant to you?
We have discovered lots of things. We learned a lot about it.
Does anyone understand how it starts?
Yeah, we do not know. I mean, we know only risk factors. In other words, like aging. Aging is the biggest risk factor of Alzheimer's disease. But before I go there, let me tell you that there are two forms of Alzheimer's disease. One is the genetic form, which we call familial Alzheimer's disease. And the other is the sporadic form of the disease. The familial form is due to genetic mutations. In other words, it comes from our genes, mutant genes. So, and people who have these genetic abnormalities, we know that they will develop Alzheimer's disease at a certain age. And we can associate the mutation with the age of onset, okay? So we have some mutations that they cause...
What are the mutations called? And what's the approximate age at which people will get Alzheimer's if they have those mutations?
There are many mutations. They're being characterized by the amino acid number. Okay, so I don't think we should go into that in detail. But what is important to know is that depending on the mutation, we can say when an individual will develop Alzheimer's disease, when the disease will start. We can predict the disease onset, the age of onset. Luckily enough, that's not very common, the familial Alzheimer's disease. It's less common than the sporadic AD, which is much more spread and much more frequent in our populations. But the sporadic AD, we don't exactly know how it starts. I'm answering your question, how it starts. The familial form, we know. It starts with a mutation. It is genetics. And those mutations are being inherited from the parents. So anybody who has these mutations, we know he has very high probabilities of developing the familial form of the disease.
Well, if mutations cause some forms of it, why wouldn't there be selection pressure to get rid of them? Why would it be heritable and continue for so long?
That's a very nice question. Okay, there are many reasons for that. One reason is that usually these mutations, they become evident after the reproductive age, okay? So past the reproductive age, there is much less pressure from development, okay? Because the individual has already reproduced or not. So that's why. So they're usually after 40. And as you probably know, we developed in days and eras where people did not live long. By 40 years, people start dying, or even 35, okay? So that time there was not evolutionary pressure. So that's, I'm answering your question. How come these mutations, evolution did not get rid of these mutations? The main reason is that. The other possibility is that we have also, mutations are being always been formed, due to cosmic rays, to oxidative factors. So we mutate all the time, but only a few of those mutations are usually detrimental. Most of them are neutral, or some of them may even be good for our survival.
Okay, so in the sporadic Alzheimer's, what age range does it tend to start?
That usually we're talking about 70 and above years of age.
And when you observe people's brains that have Alzheimer's, can you backtrack and estimate when they first started having plaque production or tau tangles? Is there any way to look back and figure that out?
Usually what brains we look at post-mortem brain. And we know that approximately from the time of the disease onset until death takes about 10 to 13 years. So usually when somebody's brain has the hallmarks of the disease, usually that individual probably had the disease for about 10 years.
30 years?
10.
Oh, 10 years. Oh, okay. So the average
10 to 13 years from the start until death. It takes about 13 years, I would say, 12 to 13 on the average.
Okay, but so when you're looking at a post-mortem brain, the person's died of it, you think the whole thing started about 10 to 13 years before?
Yes, that's right.
Well, what about onset of symptoms, though?
Yeah, that's what I mean. I mean when symptoms become overt, become clear.
Well, what I'm wondering then is, from the time that symptoms become overt, if you backtrack in time, when do you think the person first started having plaques and tangles appear in their brain? Before symptoms?
Actually, we do not know. The answer is really something that we still are not clear about when those neuropathological hallmarks become overt or start forming. Now, don't forget that many people in their 80s or late 70s they have a lot of amyloid, and even they may have tangles, but they're fine. They have no dementia. So by finding plaques and tangles does not necessarily mean that the owner of that brain was demented. About 1/3 of all aged people have as much amyloid as people with AD.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
But they have no overt signs of dementia.
Oh, okay.
So there is not really a good correlation between the amount of amyloid and dementia. That's an old problem, very old problem. That's why we cannot use only the neuropathology to say that the individual was demented, because we have many people who have such brains, but they're fine. So we need also to know whether the guy during his life had dementia, and we combine the clinical manifestation of dementia with the neuropathology to conclude that the person had Alzheimer's disease. We need both.
Okay, I understand. Oh, is there any way to image people's brains while they're alive?
Yes.
PET scan, MRI, and what's observed? So?
Yeah, we do it now with several, but they are very helpful, those technologies. MRI and PET scans and other kind of scans and other kinds of things. But again, we also need to combine that with dementia. As we said, many normal people have amyloid, but they're not demented.
Okay, so if you scan someone's brain that has amyloid and is demented versus someone that has amyloid and is not demented, what's the difference that's observed?
Well, the dementia.
On the scan itself, can you see any physical difference?
Yeah.
If you scan, let's say, 2 80-year-olds, one has dementia, one doesn't. What will you see differently in their brains?
Yeah, it means there are other factors that cause dementia that we do not see there. In other words, the amyloid that it's there, it's not causing dementia. So that means that it's more than just amyloid. That's all it means.
Well, right, but is there a pattern to the amyloid, in people that have dementia versus not? Maybe there's some differences that can be picked up on a scan.
Oh, yeah, definitely there are differences. Maybe, yeah. The problem that we have, and we're trying to clarify, is that we do not know what other factors are involved. For example, from epidemiological studies, we know that if somebody has diabetes, for example, give you an example, will have higher probabilities of developing Alzheimer's. Or if somebody has a good education, usually it has lower chances of developing Alzheimer's. Or cardiovascular problems. Usually people with cardiovascular problems also have higher chances of developing Alzheimer's disease. But these are low risk factors that you have to understand. It doesn't mean that if somebody has diabetes will necessarily develop Alzheimer's. And there are plenty of people with cardiovascular problems and they never develop Alzheimer's disease. So it's a very complicated problem because we are complicated beings. So our brain relies on thousands of different pathways and functions to function properly. So if one of those go wrong, we develop problems in cognition. So it's not that easy to identify exactly what goes wrong, because there are many, many possible pathways to something to go wrong in our brains, because they are very, very complicated. They're very complicated machines.
I've heard some people postulate that Alzheimer's is like a type 3 diabetes. There's insulin resistance in the brain cells to sugar. Have you run across that theory?
Oh, yeah, of course, I know this, but it doesn't matter what they say, what I'm saying. It matters much more what the data is saying. And I don't think there is data there to support, at least thus far, to support that conclusion. that because we have plenty of diabetes people and they do not have Alzheimer's.
Okay, so what theories are you working on? What are you postulating causes Alzheimer's?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Well, we're working with familial Alzheimer's disease. We have a model, and we're trying to find out what this model tells us about the sporadic AD and the familial AD. And we use this model because we know that AD mutations cause the disease. That we know, okay? So if we discover the mechanism by which the AD mutations cause Alzheimer's disease, we can extrapolate and we can learn a lot about sporadic AD because they are very similar, the two diseases.
Are you talking about epigenetic changes correlated with Alzheimer's or underlying DNA mutations?
Epigenetic, but we have the genetic change. We know it's change in one amino acid. One amino acid is causative. A change in that amino acid is causative. What we need to know, what's the mechanism by which this amino acid, one mutation, okay, starts the cascade that results in neurodegeneration, because Alzheimer's disease is the result of neurodegeneration. Neurons in our brains die at very high rate in the Alzheimer's brain, and we do not know how exactly these neurons die and what causes the neuronal death, the accelerated neuronal death. That's the central question of it, is what causes the acceleration in the neuronal death. It doesn't take a lot to understand that diseases like diabetes or cardiovascular disease will also will accelerate this tendency to neuronal death. Okay, they are risk factors now. They help the disease to manifest itself, but does not necessarily mean that they are causative.
Is there a pattern in the neuronal death in Alzheimer's patients? Can you observe that through scans? Does it preferentially attack the front lobe or or where does it go?
There are some neuronal areas that are more affected than others. For example, in an area that we call hippocampus or entorhinal cortex, we think that that's where the disease starts, and neurons start dying, and then it spreads to the rest of the cortex. So it's a cortical disorder. So we know that, and we know that some neurons develop what we call neurofibrillary tangles, which probably you heard of, and some areas develop amyloid plaques. Now, there were theories that the amyloid plaques is the causative agent of the disease, and if we stop the amyloid plaques or clear up the amyloid plaques, we will cure the disease. Unfortunately, this theory did not work, or it doesn't seem to be working, because we were able to clean the brain from the amyloid plaques. But as far as we know, and this is going on now for decades, we know that the disease continues regardless of the clearing up of the amyloid plaques.
What do you think the purpose of the plaques is? Do you think it's to heal, damaged areas of the brain,
right.
or do you think where do they come from and why?
Right. We don't have clear data for that either, but some people think that the amyloid plaques may be beneficial. That's a possibility. And that possibility is also the case with the neurofibrillary tangles, the tau, we call it. I'm sure you heard about it. There is a possibility that tau, by precipitating, the toxic form of tau comes out of solution. And because most of the biologically active agents work in soluble form, by becoming insoluble, they lose their activity. They become less active and less toxic, because they precipitate out of solution. Okay, so there are some people that think that that's the role of the neurofibrillary tangles. It may even be beneficial. Okay.
Huh. So, but I heard the amyloid is on the outside of the cells, and the tau tangles are on the inside of the cells.
That's right. So the amyloid, by being outside of the cell and being precipitated, it takes away from solution toxic amyloid. That's a possibility, but we do not really have clear answers yet to those. We have tons of questions. So Alzheimer's disease is still in the beginning compared, for example, to cancer. Cancer, we started in the 50s or early 60s. With Alzheimer's, we started in the late 80s and early 90s. So we need time to understand the disorder.
Are there any differences in people, again, that have plaques but don't have dementia, and plaques and they do have dementia? What about the tau tangles? Are you able to look at that and see if there's a correlation?
Yeah, there is some correlation between tau abnormalities. By that we mean the neurodegenerative tangles and the phosphorylation, hyperphosphorylation. So there is some correlation between tau abnormalities and Alzheimer's disease. The problem is that many neurons in our brain, when somebody has Alzheimer's disease, die without ever forming tau. Okay, so it's not clear that it's only tau. But as we speak now, currently, there are efforts to get rid of the abnormal tau and see if that will help the patient, the cognition. Thus far, it doesn't seem to be the case, but the jury is still out. We really cannot say yet, but I think we're going to be able to say within the next 2 or 3 years, because there are lots of experiments now going on against tau.
So the likelihood of amyloid plaques is much higher than of tau tangles, right?
What do you mean the probability much higher?
Well, it seems like in people with Alzheimer's, when you look at a postmortem brain, do you always find amyloid and plaques, or do you,
definition,
amyloid and tau, or do you find
by definition
only amyloid and not tau?
Okay, by definition, to call something Alzheimer's disease, the brain must have plaques and tangles. Okay. If the brain has somebody, let's say, has a dementia during his life, and you open the brain and there is no plaques and tangles, you say this is not Alzheimer's disease. So by definition, you have to have those pathological hallmarks to declare that the disease is Alzheimer. Now, we do have what we call plaques-only Alzheimer. In other words, some people have the dementia of the Alzheimer type, but in the brain they only have plaques. And on the other side, we have a lot of diseases, cognitive disorder, okay, that we have tangles but no plaques, okay? So, in other words, there are many different etiologies and pathways that the brain can lose neurons in addition to the presence of plaques and tangles.
Have you been able to look at the brains of people that have died that don't have Alzheimer's, but they, and if so, do they have plaques? Do they have tangles, or they'll only have plaques?
Right. As we briefly mentioned before, there are lots of people who are normal and they die at the age of 80, 85, 90, and they have as much amyloid as the Alzheimer's people, but yet they are not demented. That's why we say there is no correlation between.
But do they have tangles though? They may have amyloid and again be healthy, no dementia, but do they have tau tangles as well?
So yes, some of them do.
Okay.
Absolutely. Yes, absolutely. That's correct.
So sporadic Alzheimer's, do you think that it's still a genetic mutation that causes it? And if so, when would it arise? Would it have been with you your whole life, or just it suddenly happens when you're older, 70, 80?
Okay, no. In familial, the genetic mutations that you mentioned, in familial Alzheimer's, definitely, the genetic is very clear. The Alzheimer's disease is caused because of the mutation.
I know you're born with it, but again, in sporadic Alzheimer's,
yeah,
is there a genetic mutation that's responsible? And if so, when does it arise?
Okay. No, it's not one or two, but the genetics may still be very important, because we have observed that people with relatives that had Alzheimer's disease, these people have higher probability of developing Alzheimer's disease than people without relatives with Alzheimer's, okay? So apparently the genetics is there also, but it's complicated. There are many genes. It's a genetic or polygenic disease that many genes together contribute, okay? So that's why the genetics of I'm talking about the sporadic AD now. That's why the genetics of the sporadic AD is very complicated. And in the contrast, the genetics of the familial AD is very simple. As you said, it's one mutation. In addition to that, in the sporadic AD, we have what we call polymorphism, which means there are somehow normal variants that are in the population, and some form of these variables also seem like promoting the disease. Because if you examine Alzheimer's disease genetically, Alzheimer's disease patients, you'll find that people that have this kind of variant have higher level of Alzheimer's. So there are normal genes, in other words, some variations of them that they contribute to the development of the disease. But apparently that's not enough, because there are many people who have these variants but still do not have Alzheimer's. So there are other factors, and that's what we're trying to find out. What other factors are there? That could be diabetes, could be heart diseases, could be cancer, could be lots of different things.
So what's the difference in someone that has sporadic versus familial Alzheimer's in terms of their symptoms and their progression? Is it faster, slower, different?
Okay, there are some differences. There are some clinical differences. The neuropathology is very similar, but clinically they are not identical. For example, the familial AD is usually more toxic. In other words, they die sooner than it doesn't take 15 years. It takes probably less, 10 years or 8 years. So they are more toxic in general. And there are also changes, differences in depression and other kind of variables. They're not exactly the same, but in general, they can be classified as Alzheimer's dementia, and the neuropathology is very similar. It's plaques and tangles.
Okay. I just didn't know if one progresses faster, but like you said, the familial is more toxic. So what does that mean? It just the progression is faster,
exactly. The progression, it's faster. The progression.
A lot faster, or a little faster.
Yeah. No, it's measurable. We can measure it, and we can conclude that. Therefore, it's measurable.
Okay, gotcha. Is there any difference that you notice in Alzheimer's in men or women, or different ethnicities? How it affects them?
Yeah. I mean, there is some evidence, and that's epidemiological or statistical evidence. Okay, I want to make that clear. There is small difference. It's the same way that the people with education seem to have a slightly lower probability of developing the disease. It looks like women may have a little bit higher probability to develop the sporadic AD. But again, the difference is small and depends on many other factors as well. So it looks like the females may have a little bit higher probability of developing the disease.
So what specifically are you researching right now? What's the latest and greatest?
The mechanism by which the FAD mutations cause Alzheimer's disease. The mechanism by which the FAD mutants promote neurodegeneration. Because as we said, the causative situation is the neurodegeneration, is the death of neurons that causes the problems with cognition.
Well, what can you do? Can you use like the special fluorescent glucose and do a brain scan, or how can you get to the cells in the brain to observe?
That's right. Yeah, we're doing that also. That because the brains that suffer from a disease have different rate of metabolism than the normal brain. So we can measure it also. The same, another variable that we measure, but all of them are not really 100%. There is always caveats. The other thing is that the Alzheimer's disease brain is smaller because it loses mass, and you can see that in a pixel. You can by using MRI. Okay, so that is the other methodology that's been used. Mass of the brain. So we have various techniques.
But can you look at the transcriptomics of these cells, or the?
Yes, that's correct.
How do you get to these cells to observe them?
We call it transcriptomics. We try, in many laboratories, and mine, we are looking to find out whether the transcriptional differences between in the neuronal cells of Alzheimer's disease brain and in neuronal cells of normal brains. We want to see whether there are constant differences and measurable differences in the transcriptome, as we call it. In other words, in the transcription. And by that we mean that the operation that makes proteins from genes. Okay? So we call that transcription is involved in how the genes is being translated into proteins, because the proteins are the workers of our brains. They're doing the work. Okay? So we want to see whether this very important operation called transcription is being affected differently in AD than in normal. And that is a very, very difficult and time-consuming operation and costs lots of money.
Okay.
But you're right. Transcription is very important. The genomic is important. Very few neurological disorders of the brain, by the way, we know how to cure. There are many. There are a lot of different cognition problems, and cognitive disorders. We just started. It's going to be a long way before we really understand how brain cells die and why they die differently in different diseases.
Are there any biomarkers, anything that these cells will shed into the bloodstream that can be picked up and observed?
Yeah, sure. There are some biomarkers like phosphorylated tau, for example, maybe neurofilaments. Yes, we follow them. That's correct.
Okay, well, very good. Well, Dr. Robakis, where is the best place for people to go to learn more about your research?
To my NIH grants and my papers. Also at Mount Sinai, we usually have a website.
Okay.
And we say there what we are doing.
Excellent. Well,
It's all public, but it's esoteric. Somebody has to know some form of biology a little bit to understand this. So for the people that are not experts, it looks like difficult to understand. And of course, that is expected.
Okay, well, very good. Well, thank you for coming on the podcast. I appreciate it.
Yeah, sure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.